So I think we can start, and like I mentioned at the beginning when I gave the introduction of the afternoon panel and the workshop, this is funded by the NSF. It's a 2 years funding that NSF provided to cover both last year and then this year activities. You just heard 2 talks from Nico and Micah about different approaches to optimize media formulation for specific needs. We're now going to move into what we call a panel discussion. So this is an open forum, and it's a joint open forum where we're bringing a member expert, both from the plant and the IVACS section. So what I'm going to do first, and then we're bringing also back the 2 speakers of this afternoon. What I'm going to first do is to ask the panelists to quickly introduce themselves, and then I'm going to open the floor for questions. So this is really kind of an expert corner that we created where you can ask questions of interest, and then the panelists can bring their own experience and expertise to address your question. So we're going to start with John, if you don't mind to give a quick intro by yourself.
Good afternoon, John Harbell. My area of interest is cell and tissue-based bioassays. Oh, sorry.
Well, that was quick. Thank you, Don.
For regulatory purposes.
My name is Greg Phillips. I started work on plant tissue culture back in the 1970s. In the 70s and early 80s, I did a lot of work with basal medium manipulation for legumes, particularly for clovers and soybean. That resulted in an L2 medium. I don't use it for everything, just legumes. Later on, I got involved in onions and peppers and found that MS basal medium was not adequate, especially for the onions, and discovered that some onion researchers, Dunstan and Short, had modified the B5 basal medium. They called it BDS, B as modified by Dunstan and Short. And it had a little more nitrogen in it, it was a little better buffered, and it worked better for onions. And I've worked with woody plants, conifers, shrubs, cactus, clovers, grains, weeping willow, corn. I've worked with all kinds of different plants. And MS is great for some of them, but it's awful for others. And so my lab also developed another modification of BDS called BABI, which we found to be highly suitable for virtually every plant species we tested it for. Then in contrast, in recent years, I worked on soybean microspore culture, which requires less nitrogen and less sugar, nitrogen starvation, so it's another basal medium manipulation. And so with that, I'll pass to my friend Guy.
Thank you. I'm Guy Smarre. I'm in the field of entomology, and my experiences on insect cell culture and tissue culture go back to more than 25 years ago, to culturing imaginal wing discs. So when you bring them in culture medium, you can develop small wings. So in the early 90s, I was, let's say, culturing them as a bioassay to investigate the growth and development, because these imaginal discs, in fact, they generate the adult appendages of the insect, so like wings and legs, and this is a process of metamorphosis. Now, to develop this, there was not much available. There was Grace's medium, but I had to optimize, let's say, ingredients, growth factors, hormones. So I could realize this, growing small tissues, and let's say from an adult stem cell cluster to small wings with scales. They didn't fly away, they stayed in the culture medium. But that was a kind of optimization, small scale. Then I continued with immortal cell lines from insects, also from insects. By the way, insects have a fantastic property. You can culture them at 27 degrees. So no problems about pathogens that grow at 37 degrees, no requirements on carbon dioxide. So this is making life a little bit easier than with vertebrate cells. Also, that field has changed a lot. When I started, there was a lot of culture media, a lot of different culture media. You also had to add quite a lot of ingredients. And then there was a boost of the field where serum-free medium became available. So in the past, most of the people were using 5 to 10% of fetal calf serum. But then the serum-free media came, which was making life more easy. But on the other hand, also let's say the ingredients of the medium were not always shared by companies. What is my interest looking to the future? I think when we look to cell culture in small and large scale, I think use of biosensors is very interesting to do an online monitoring. So also this kind of feedback control systems, as we also saw in the presentations before the break. I think these are really optimizing production systems. So real-time adjustment of pH, nutrients, and let's say also oxygen. What is also, I think, interesting, I think this is also a growing field, is the use of AI, not only to explore literature, but also to, let's say, to manipulate or to employ the very complex biochemical pathways that are in plant cells, animal cells. So just to understand where you change something, what is happening, because biochemical pathways, they are so complex and there is a lot of action and reaction and feedback systems. So I think using AI in combination with literature, this will really help in neural network thinking, which I think is maybe beyond our own capacity of our brain. This goes, of course, together with the enormous amount of information available in the literature in omics at level of genes, RNA, and proteins, peptides. So this is a little bit in a nutshell where I come from, what I did, and what I see as important directions. Thank you.
I think they have the mic. Yeah. Oh.
Thank you. So I'm Nico. My background is in agricultural and biological engineering. I've been with 4A Bioscience for a little over a year. And before then, I got experience in both greenhouse production, vertical farming, broadacre, and microbial ecology.
Thank you. Yes, hello again. I'm Micah Stevens. I did my undergrad degree at University of Kentucky in horticulture science, and then went on to Purdue University in the forestry department to study forest genetics with an emphasis on tissue culture and clonal propagation of eastern black walnut. I've been doing woody plant tissue culture for about 16 years now, close to 8 of which has been in a commercial setting. Yes, I really like plant propagation, specifically tissue culture, and just trying to understand, better some of the factors that control woody plant in specific tissue culture. Long-time panel attendee, first-time panelist, so excited to be here and happy to answer all your questions. Thanks.
Great, thank you everybody for the introduction. So again, as you can see, the panel that we put together is a panel that has a different background in terms of application, and also a different way to and how address specific challenges associated with the specific explant and tissue for specific applications. So I'm going to open the floor to questions. As usual, if you have a question, please raise your hand, say your name and where you're from, and then we can start. I already start to see emotion there. Yep.
Hey, I'm Ken from Oregon State, and So for my PhD, I'm working with somatic embryos in grapevines, and one of the major bottlenecks is converting the embryos into mature plants. So I was wondering if you guys had any insight into the role of like 2,4-D in embryo maturation process, as well as ABA, and also any other factor like maybe pH or nutrients.
Okay, I don't know who was Craig. Do you want to get a start?
That's a complicated question. So grapevine, and you're getting somatic embryos, but they're not converting? Are these different genotypes then had been used before that have been widely published out of Florida? And
This is a pretty well-established problem in grapevine, is that you'll get a lot of like weird mutant phenotypes in somatic embryos that have trouble converting into plants.
Okay, so there's some abnormality in the formation of the somatic embryo as well?
Yeah, exactly. There's like
So
fusions or...
you must have suboptimal conditions.
Right.
Or pathogens.
Pathogens. No, this is not uncommon in certain plant species. The protocols may not be as optimized as they could be, and so an imbalance of hormones for that particular genotype might result in a different phenotype than you do in another line that you have cultured. I don't know if Wayne Parrott's in the audience, but he worked with soybean somatic embryos, he and John Feiner, for a long time, and that soybean is an difficult plant to convert from somatic embryos. Feiner could scale them up and fill this room with them if you wanted, but then getting them to convert to plants was more of a challenge, and Wayne could speak to that, how they changed the sugar from a glucose or a sucrose and changed it to maltose, which is slower metabolizing. They may have used activated charcoal to help get rid of maybe some toxic substances or an excess of auxin. So those are polyvinylpyrrolidone and agents like that sometimes help. Benzyladenine does have a bad rep of causing some abnormalities in both somatic embryos and in proliferating shoots, organogenic shoot cultures. If the level is wrong, that will happen. 2,4-D is well known to inhibit maturation of somatic embryos. It's very strong in that regard, and other auxins may not have that same inhibitory effect. For example, picloram tends to provide cultures that can sustain regeneration of plants for much longer periods of time. Like in onion, for example, going from 9 months to 3 years was the difference between using 2,4-D as published versus using picloram that we optimized for it. So it may be a basal medium problem or a physical environment that needs addressed, or it could be the balance and source of the hormones.
Thank you so much.
Just quickly add to that with my experience, definitely not too experienced with somatic embryos and not in Vitis, but in the jugular somatic embryos I've worked with, running into issues with germination. We've sort of overcome this with a chilling period's really worked, and even with walnut adding a little, GA3 or GA4+7. But I think my biggest advice would be, have you reached out to David Tricoli at the UC Davis Plant Transformation Center? Okay, yeah, he would be your best person there. He seems to be the expert on all things grape somatic embryos.
Okay, thank you for addressing this first question.
Hi, it's Lucas van der Zee from Wageningen University. I am wondering, I read somewhere once in a paper as sort of a naive first approach to creating a plant tissue culture medium to look at the elemental composition of a seedling or of a regenerated shoot, and use that as a basis of your nutrient composition, of your basal salt mixture. Is there any link known between the elemental composition of a regenerated shoot and the optimal composition of a medium and of the minerals in a medium?
Have you read the Murashige and Skoog paper from the 1960s? That's
I
all that
haven't.
paper is about.
Yeah.
What did they start with? Plants. They did an elemental composition of the tobacco plant and used that as their starting point, and then optimizing and developing the Murashige Skoog formulation, which they felt was optimized for tobacco. It is very hard to beat MS for growing tobacco, although BABI works just as well. And it starts with a different set of minerals. So one of the things you have to worry about if you're doing basal medium manipulation in plants is, what set of salts are you going to start with? Because that's what you're optimizing. Unless you start with an initial panel with different salt sources, like DKW versus MS versus BDS or BABI. Those give you some quite different results because of the different salt sources they used. And based on those responses, you might decide DKW is the best to start optimizing and finding out what's best.
And so this worked for them in tobacco. And do you feel like it's in general true that if your medium composition closely resembles the elemental composition of the plant,
No.
that it will perform better?
Murashige would not have had a thesis if that had been the case, because their starting point based on initial tissue elemental analysis was not in the same proportions as they were in the final MS formulation. So therefore, there's not a direct correlation, and I'm sure mechanisms of uptake, for example, mineralization, all that kind of stuff probably plays a role in that.
Yeah.
You might not see a whole lot of phosphorus in the tissue, but by golly, it is recycled and recycled and recycled throughout the life of the plant.
Yeah. All right, thanks.
Hello, Claudia Garcia here from Gali. I have a question about media optimization. What is the best approach to do a media optimization, since we know many interactions are happening between the organic elements and also the nutrients in the media? For example, if you have a source of sucrose and this is completely depleted, you are not going to see that the plants are going to consume the other nutrients because there is a limiting factor here. So how we can approach the best this research to not lose information when you are not measuring completely the whole picture?
Sorry, could you maybe repeat the question? I'm not sure I'm...
Yeah, that I mean is when you are doing an analysis or a media optimization, which is the best approach to see all the balance of the nutrients in the media, because it's not only the macro or micronutrients that are going to influence in the growth, it's also the organic components. So if, for example, sucrose is completely depleted, there is a limiting factor that is not going to allow the cell's growth or the plant growth, and you cannot measure if it's the nutrients or is the sucrose or what it is that is limiting the growth in the studio.
Gotcha. Okay, I think I understand. And if I don't answer your question, please feel free to chime in. So for the majority of the problems that we run into, when the production lab comes to me and says, Micah, this cherry rootstock is not growing like we expect it should. All the other cherry rootstocks are growing on this media. This one's not growing. What's the issue here? then my first thought is not going to be, okay, I'm going to go change the basal nutrients around. It's potentially something else. Yes, so I'll look at the carbohydrates first. Maybe it needs to be switched from 30 grams of sucrose to just 30 grams of glucose, and if we run into hyperhydricity, we'll deal with that. There's other things you can add to compensate for that change in osmotic potential in our media, or it's the gelling agent. What I didn't show, but what we did was these same nutrient uptake analyses at using different gelling agents. Either 8 grams of agar versus 2.5 grams of Gelzan. And what we saw, these gel and gum based media were able to take up the nutrients more quickly and at higher concentrations than plants grown on just agar. So that's why I think looking at your carbohydrates and your gelling agents are a better starting place than jumping straight to thinking that you need to retool your whole basal nutrient formula. If you try your carbohydrates, that's not working, gelling agents plus carbohydrates, that's not working, then I go down the path of tweaking the basal nutrients. So yes, I think it's really complicated to try to do them all at once, and it's a little bit easier and less time consuming to look at the organics like the carbohydrates or the gelling agents first. Does that answer your question?
If I may expand the question to the rest of the panel, because your question is specific to plant, correct? What is your take, John or Guy, when it comes to media modification in general? What is your starting point or kind of the best practice that you always kind of follow, or you recommend?
Oftentimes you have an established medium, and again, I work in cell-based cultures that are used for regulatory toxicology, gene tox and so forth. But how they're handled in the laboratory can make a tremendous difference. And an example is a Fisher's medium, which has been used for many years for lymphoma cells, but it's very light sensitive. And so if you have white lights in your laboratory, as most people do, white lights in your hood, you can denature that medium and render it unable to support the growth of those cells in about a day. So if people put the media bottle in the open water bath, go off to lunch, come back and feed the cells, do that a couple of times, that medium may no longer be active. So that's one of the things just to be aware of is what Dick Ham used to call the gestalt tissue culture. All those things that could impact the cells and the medium are really important. The other is filtration. You take a medium and you want to add a growth factor, or you want to add something to it, and you filter sterilize it. Now, we're more concerned about that at 37 degrees than you might be in other situations. But the serum-free media often have components that are colloidal, and iron is one of those for the keratinocyte medium. If you filter that, you strip out the iron, and that medium is no good whatsoever. So it's things like that to be aware of that might complicate taking a basal medium from someone else and making it and using it in your own situation that then doesn't work as well.
Thank you. So I'm Jose from KWS, and I was wondering, what do you think are the major challenges on implementing these AI technologies? So for example, the random forest, machine learning models for optimal media composition. Because the way I see it also is you spend a lot of time collecting like good quality experimental data, but this might be very genotype or like crop dependent, right? So I was wondering if you know like how to solve this situation.
Yeah, I can jump in there. So I think the first thing is you need to have your experimental design and data structures set up in a way that can be fed into these models. That's obviously the first step, and then once that's there, you can interchange which one of the models you're using. I think the second part of your question is essentially how can you learn from one genotype to the next. Is that your question? Yeah, so there's different techniques for this that essentially allow different runs to learn from each other. So you have like, you do 2 genotypes at first, you get your outcomes there, and then those learnings can be incorporated as prior information into the later trials. So that's how I would approach sort of the diversity that you're going to be dealing with on a genetic basis.
Just my 2 cents, if I may. I think that the more that we're able to understand the nutrition requirements for more crops, the more that we can feed into these models, the better off it's going to be, and the greater breadth of knowledge we have is going to help direct new decisions on new genotypes. Filling out that clade of relatedness between crops. Is it family specific? Is it just genus specific, or is it just species specific? And the more we can fill that out, the easier I think it's going to be to answer those questions.
And I'm going back to the floor, but I like a comment that Guy, you made at the beginning of your intro when you mentioned the use of biosensors as becoming more and more valuable, for example, to generate data that can be fed into this AI model. Is that what you were kind of mentioning at the beginning? I think that's something to keep in mind when it comes to creating data that can then be fed into these kind of models. Is that the direction where you were going in your comments?
I think this is something that you see really at mass production of cells, where you have biosensors that can do very quickly monitoring. As I think we also saw the example before the break on this evaluation of phenotypes, these colors, where it is allowed to do an evaluation very quickly, much quicker than before. So these kind of biosensors allow you to do, let's say, a judgment and feedback system, together with, of course, quite a lot of knowledge about the biochemistry, the biochemical pathways, and literature. So I think this is really exciting when you see that this kind of AI, when it's integrated with literature and biosensors, that you can really understand and respond relatively quickly. I think this is in all fields. This is not only in plant or animal field, but I think we can learn a lot from this. This kind of automatization and also objectivization. Of course, as we heard the question about different cultivars, or sometimes maybe the difference between cultivars within one species is bigger than between species. It's all depending on, let's say, what is your genetic or biochemical background.
Hi, Sarah Eubanks, also from KWS Germany. Gregory, you mentioned you worked with soybean microspores and that they need this nitrogen starvation. This is known for a lot of crops, that they need this kind of nutrient starvation in order to further develop. I was wondering if you could use this this nutrient uptake cases to really study not only what you need, but what you don't need at certain points of development in order to induce this embryo induction or for callus induction. If you can use this somewhere on a single cell level.
Well, that's a very good point. There are things that are inhibitory to the next step, just as there are things that might be stimulatory to get to the next step. And when I think about one of my students was working with a conifer callus tissue, long term, and she found that she had to do a lot of back and forth. Give it a signal and then take the signal away. Let it calm down, so to speak, give the signal again. Sometimes it took multiple rounds of hormone signal, nothing, hormone signal, nothing. Now with trees, this might make more sense than it does with crop plants, if you think about how trees grow, etc. Trees tend to be very phasic. They have a spring flush, for example. They have fall abscission. So I think about those kinds of things. There's probably something that is inhibitory in that step. We couldn't figure out what it was, but Heather went around it and found another way to get to that next step.
Maybe just adding to this here, it's based from my experience on insect cells and insect tissue culture. More does not always mean better. I mean, more in concentration or more nutrients does not always means better. And I think what Greg is also saying that is, think about how the plant or the animal or the insect is growing on your tissue that you work or your cell that you work with. I mean, look to the biology of your source and understand this. I give you the example of imaginal wing discs where there is growth, but not with an extremely rich medium. So you need a kind of a minimum essential medium, and then you need a pulse of a steroid hormone to get started with the development. When the development started after the pulse of the hormone, then you need nutrients for, let's say, cell replication, cell stretching, and so on. So it really demands a better understanding about the biology, how a tissue or an organ is growing, and then you can simulate this. I mean, it's very basic. It's very logic, but yeah, because everything is so much easy with kids and all these cultural media that are commercially available, people lose a little bit the knowledge about, okay, how to do this. So sometimes you have to go 2 steps backwards and think about, okay, what is really needed? And sometimes I advise my students to start with a minimum essential medium and just add things, what you think is essential, depending on your biological question, on what you want to realize. Whether it's growth, whether it's an immune system, whether it's production of an antibody, whatever. So it's really depending on your situation.
My name is Sandra Romero. I am from Verinomics, Connecticut. And my question is related to if you have recommendations for the transition, regarding the media composition and the transition from media to soil. I would like kind of to know if you have any insights about how to do this transition easier for the tissue to the artificial media or conditions that we are providing to the plant, to the real conditions in soil.
Are there any specific crops that you had in mind?
Well, we work with different crops, but dicots, monocots, in general, not necessarily a specific crop.
I think, one of the biggest factors for, optimizing anything that we produce at the nursery is increasing our survival rates. Because an increase in survival rates in the acclimatization tunnels saves all that much time and labor in the tissue culture lab. And what we've found is that a couple of things. So for most crops, what we used to do was to induce roots. They'd either go in the dark for a couple days or sit on the shelf for 4 days, and they'd go out to the tunnels and get planted in soil with no roots on them. And this led to not great survival. So what we started to do was let them sit in the light, so an in-light root induction for 12 to 15 days, and then kind of measure when the majority of roots that would come out. So we'd do these trials, and we could say that, okay, by day 18, 90% of these are rooted, and we can see visible roots, but we don't want to break those roots off while we're planting them, so we'll plant them on day 16, knowing that they're only going to be in the tunnels for 2 days without roots on them, for the most part. So that's one thing that we do to really increase survival, is not planting them before they're ready. Because I mean, the leaves that are made in tissue culture are basically non-functioning. The stomata don't really work. They're not suitable for photosynthesis and all that. So we need to nurse the plant along for as long as we can before it starts to put on new growth and make new leaves. And one of the other things that we've done with our walnuts is that we'll actually get these Jiffy plugs. I don't know if you're familiar with Jiffy plugs. They're just compacted media that's sterilized. We'll induce the walnuts to root, put them in the plugs, let them root in the lab for about 2 weeks until the roots sort of penetrate and get through the plug, and then we move them out to the next step, which is the greenhouse, because we found that as soon as these microplants have roots on them, they're a lot more robust to handling the stress of moving to ambient greenhouse conditions. So I think the biggest thing was minimizing the amount of time that they're in ambient, outside tissue culture conditions without roots on them. The best you can do to ensure that they're rooted before they go to sort of acclimatization tunnels is what we found to really work for our crops.
I'll add a little bit to that. I agree. From my experience, we always preferred to have plants rooted before we took them to soil, although there are some species, particularly woody species, that are often mist-rooted in a greenhouse. But if you're not working with one of those species, it may be more of a challenge. The other thing that I would point to that Mika didn't mention, or I didn't hear, was depending on the plant, the rate at which you expose it to ambient air can be extremely critical. A cactus, you can probably just take it out of the petri dish or tube or whatever you're growing it in. If it has roots, throw it in soil, it will survive. But most other plants won't do that. The leaves are more likely to lose water very quickly than a cactus does. And so you really have to, the first few days of trying to transition from a petri dish environment, air environment, to ambient environment, that transition is very critical.
I was also going to add, I think one way to think about this as well is to kind of break it down, like what's different when you're going from tissue culture into soil, and right, like Micah, you mentioned lighting. There's obviously like the soil physical environment is different, but one of the other characteristics is the microbial environment, right? You're going from a sterile to all of this new life, and so something that I would think could be helpful as well is to look at seeding either the roots or some other part of the plant with some beneficial microbes.
Yes, and I can confirm this. We've done this with pecans, and we found that getting pecans to survive and acclimatize well using some sort of endophyte in the soil mix seems to help them survive and continue to grow.
Yeah. Hi, my name is David from Phyton Biotech in Germany, and we are working also with these plant basal mixtures. And have you made any experience with potential impurities of those or in those individual single nutrients in those mixtures when you have adapted your cell line to a distributor, for instance to Schäfer plant media or Phytotech, and then your technician forgets to order new and you have in your shelf another one. It's identical on the piece of paper, but after 2 weeks you do see any differences in changes of maybe morphology, color of the cells, cell growth. Have you seen something like that?
Yes, 100%. Not as much with nutrients, definitely with agar. Currently, right now, we're fighting a bad batch of agar that refuses to solidify. But with nutrients, we tend to have single source suppliers and have not noticed too much differences batch to batch. But again, for that reason, we tend not to switch suppliers unless we have to. I know that when we were, during COVID and supply chain issues, we were having to switch up suppliers and we were noticing differences, but it was not enough. We're dealing with the more robust systems than cell lines, so we can kind of take the hit a little bit there, and because it wasn't enough to seriously impact plant growth. But yeah, absolutely, we do see differences in batches in that.
Hi, Ariane Tremblay from Green Venice in Davis, California. So coming back to AI and predictive modeling and things like that, so if we were to have a company that wanted to invest the time and the money to capture, as many information that is available in public database and, all the research that everybody has done, do you believe that we have enough information to actually, as of now, to actually build something that will be as predictable as possible? Or we should chase more?
I can jump into this a little bit. I think there's two sources of data essentially that exists out there, right? There's everything that's been done, that's been published, and then there's actually more sort of these like private data sets of what your lab or what other labs might be working on. And I think one of the big opportunities is to combine, right, as a lab, what are you doing in-house and what else has been done in the world. I think the other interesting piece here is we don't have to only do black box machine learning models, right? Like there is a lot of structure, right? Like we've been talking about media. There are some other things that are at play, but when you're modeling biological systems, it's possible to give these tools, the modeling tools, advanced information, and not just like take a sea of data and try to learn from the data alone. And so I think the possibilities there, a lot of them come from blending private and public data as well as giving the models some knowledge to start with.
Yeah, hello. My name is Alperen. I'm working with Dr. Peret at UGA. So since we have all of you here, I want to ask a general question based on your experience and given your history in tissue culture in general. So if you are given 3 main components that you should list by the priority, like the most important 3 factors in culturing or optimizing media conditions in your own experience, and what kind of things you will list, like the PGR, the concentration, light intensity, temperature, or like anything that comes to your mind, because there are so many variables. So in your experience, which ones are the most important 3 components of a successful plant tissue culture? Thank you.
Okay, who wants to start? And I will expand to all the panelists.
I'll kick it off, but I'm not sure I can keep it to just 3. I'm not even sure what my top 3 are, but a bunch of things come to mind. For one thing, learn from an expert.
Yes, I just want to hear if you guys will tell different orders based on your experience. That's the reason I asked.
That's the first thing I'll say is learn how to make media from an expert. I mean, you can pick up the literature and you can imitate what they've done, but you probably will not have all of the ins and outs of how to make specific stock solutions, because if you put these 2 things together, you get a big precipitate in your medium. So you have to separate some things. You have to know how to solubilize these things. We all use EDTA or other chelating agents. They don't stick just to the iron or whatever you thought you put it on. After it's in the medium, it starts going out and finding the zinc and the cobalt and everything else too. So you have what you start with and then what ends up after you've either filter sterilized it or cooked it. And that's what you're actually feeding the cells. So that was a half dozen things at least in there, right? So somebody else can offer.
Well, Dick Ham, again, going back to him, he would take a component and he would optimize for that, and then he would start the next component, and he might have to change them. The other thing he did was to make his components in individual batches that were appropriate for solubilizing and sterilizing that particular material. So in the 1980s, you could buy keratinocyte medium from, it was 12 bottles, some frozen, some not frozen, all sterile, and you made your own because you could not put them all together and then sterilize it.
Yeah, I would say, like, to begin with, you want to look at basal salts and carbohydrates. I would agree with kind of your talk earlier on that, Micah, and you know, things like hormones come next, and then, you know, you have ingredients for various specific purposes like activated charcoal and various other things that sort of come later on.
Okay, so just to make sure I'm understanding your original question here, you're wondering what my Mount Rushmore of media components might be?
Yeah, or the conditions in general.
Okay, it's a great question. I would, and again, this is just my experience working with the crops that I've worked with in, you know, sort of a commercial lab setting. It's your, you know, nitrate to phosphorus ratio, your ammonium to potassium ratio, gel and gums. That's, what am I at, 3? 4, I'm gonna say glucose, but a close second would be to don't sleep on the cytokinin 4-CPPU. It's kind of an alternative to TDZ, and I have not seen it nearly to have as, you know, the deleterious side effects you see with TDZ. But 4-CPPU has seemed to be, you know, really useful for a lot of our crops, where, you know, other cytokinins kind of fall short, and TDZ leads to, you know, wonky morphologies. So that would be my Mount Rushmore.
Well, along those lines, once you have your basal medium set, you also need to look at your sources of auxins in particular, because most of them are synthetic and have unique properties in addition to their auxin properties. So for example, everybody uses 2,4-D. They just throw a glob in there and expect callus to form and somatic embryos to form. And then they remove the 2,4-D and expect the somatic embryos to develop, which, as we discussed earlier, doesn't always happen, even in so-called easy plants. So, for example, when I discovered picloram as an auxin, I found I could scale that one to the precise concentration that would give me very discrete developmental responses in clover in particular, but in other plants too. Whereas if you take a 2,4-D, I bet you would have great difficulty getting very specific concentrations to elicit very specific responses. It does what it does almost regardless of the amount you use. Okay? So some people used to argue about cytokinins as well. What, you know, BA causes hyperhidricity, and so you don't want to use. I see less differences among cytokinin sources than I see among auxin sources in that regard, although there can still be differences.
Okay, thank you very much. I'm Bakala from Weizmann Institute, Israel again. As you know, silver thiosulfate, STS, which is an ethylene action inhibitor, has been shown in some plants to induce multiplication. There is an example a long time ago, I don't remember exactly the journal, but there was a publication about its use, and there are other places where this silver thiosulfate has been used, especially for organogenesis. Do you have any experience in this with the use of especially conversion of somatic embryos? In addition, it has been known also from its negative effect on the environment. I don't know what its acceptance today is, if it is really being used in the tissue culture industry. Thank you.
I do not. Sorry.
we're not clear of the compound. It sounded like SDS.
Yes, STS, silver thiosulfate, ethylene action inhibitor. It's being used in the post-harvest of cut flowers, but also in tissue culture.
See, we talk about that as making shampoos, sodium lauryl sulfate.
It is STS, yes, silver thiosulfate. Silver thiosulfate, STS.
I have tested silver thiosulfate in a couple of different plants, but you do not see it appear in any of my publications.
He doesn't use it. I think that's his way to say that he doesn't know.
The one time I have used STS was we've been propagating this ornamental tree, Parksonia florida, a blue palo verde tree, and we're having, it's this pinnately compound desert species. It was suffering from a significant amount of leaf drop in tissue culture. We started adding STS, and I think the inhibition of ethylene has really mitigated leaf drop in these cultures. So it's helped with premature defoliation in this one specific crop that we do, but that's really my only experience with STS. Cool, thank you.
Thank you for that comment. We have one question.
Yes, hi. I'm Manuel Gastelbondo. I'm part of the blueberry breeding lab at the University of Florida. And I wanted to ask about somaclonal variation. Even when the conditions are ideal and you treat the plants correctly, in theory, you're subjecting the plant cells to very fast propagation, and of course there's an accumulation of somaclonal variation, mostly epigenetic, but also in the genome. So I wanted to know your experience with regards to how often do you think, I know it depends on the species, but to reinitiate the material, and also if you use any technique to monitor this somaclonal variation to address this period of reinitiation of refreshment of the material. Thank you.
Great question. I can start off real quick, because this is something that's near and dear to every nursery folk. Selling off-type trees is a sure way to not make people happy. So we do everything we can to avoid that. And when it comes to what we propagate in our tissue culture lab, is that we stay ahead of the curve on it by reinitiating about every 36 months, 3 years. We're going back to a clean plant center, foundational source, getting that material, bringing it in, reinitiating it. We run it for maybe 2 production cycles. It'll take about a year to initiate and build up, run 2 production cycles, and then it gets phased out. So we, stay ahead of the curve in that regard, and we also try to, limit these, cytokinins that we're using. Whether that's helping, it's not sure, but we don't observe any off-type phenotypes. But then to the second part of your question, I think monitoring somaclonal variation is incredibly difficult. Because you could not show up for years down the line, at which point for us it's already out of the nursery. If it's luckily, if we're lucky, it's something benign. If not, then it's something really bad that's going to impact the orchardist, and that's what we don't want to happen. So there's nothing that we can actively do to quantitate changes in somaclonal variation. And sort of what I've seen, there's some studies in changes in the methylenome throughout different culture periods. I think there's actually a study in avocado, right? I think maybe, or Yeah, anyway, so looking at how tissue culture is affecting changes in the methylome, but actual semiclonal variations, I'm not sure, and we just do our best to stay ahead of the curve on that. So that's my experience with semiclonal variations.
Okay, so I think we are close to the end of this panel discussion. Before we leave, first of all, I would like to thank again our panelists for tonight. And then I'm inviting all of you to keep the conversation going on the floor. You have multiple days to keep interacting with the panelists and also among yourself. This is exactly one of the benefits of meetings like ours, where you have plenty of opportunity to really talk and connect and share experiences. Before we leave, I just want to quickly remind you that at 7, from 7 to 9, we're going to have the welcome reception in the main ABCD room. That is the room where we're also going to have the posters. And tonight we're looking at the event poster numbers. And then if I have students in the room, a reminder that at 8:30, from 8:30 to 10, in the Granby ABC, you're going to have your student and postdoc networking opportunity. So again, from 7 to 9, the welcome reception, and it's also an opportunity for you to start to look at the event poster numbers. And then if you are a student and postdoc, networking from 8:30 to 10 p.m. Otherwise, we'll see you tomorrow morning. Again, we're going to start at, I should know the program, but I don't know the program by heart, 8 o'clock. And tomorrow is going to be a plenary session that is about biotechnology approaches for animal and crop improvement and environmental risk assessment or genetically engineering organisms. That is kind of a follow-up of the panel discussion that we had this afternoon related to stewardship. So thank you again for joining today, the different panel and workshop, and thank you for attending the 2025 In Vitro Biology meeting. Thank you.